Okay, hello everyone. Welcome to VSPG. Today we're hearing from Dr. Romain Guilbaud. Sorry, please. Is that correct, Guilbaud?
Yeah,
All right,
sure.
all right. Romain Guilbaud, and on phosphorus in the Proterozoic. I'm going to go ahead and introduce Romain for us. So Dr. Romain Guilbaud is an isotope geochemist interested in the chemical evolution of Earth's surface and its co-evolution with life in deep time. He combines fieldwork and analysis of ancient material with experimental approaches to better understand complex mineral interactions during deposition and diagenesis. Roman's work has revolved around redox-sensitive mineral formation, such as sulfides and oxyhydroxides, and their impact on their environment. Also on understanding non-traditional isotope systematics during diagenesis, and on bioessential nutrient cycling through time. Roman received his PhD from the University of Edinburgh in Scotland, conducting experiments focused on iron isotopes during sulfide formation. After his PhD, he did several postdocs in Newcastle, Leeds, and Cambridge in Precambrian biogeochemistry, doing both fieldwork and experiments. After a lectureship at the University of Lancaster in the UK, Roman began his current position as assistant professor at CNRS in Toulouse, in France. We're looking forward to hearing about the phosphorus cycle today, so please take it away, Roman.
Thank you very much, Alex. And thank you, Andre and Alex, for this invitation. I'm delighted to be with you this morning or this evening, depending on where you are. So yeah, I don't do only isotopes because obviously I'm going to talk about phosphorus today. And I thought I would gather data acquired with PhD students and collaborators along the past 10 years, I would say, something like that, with an experimental approach to how can we reconstruct the phosphorus cycle in the Proterozoic. Obviously, the Proterozoic is where many things happened, including the oxidation of the Earth's surface, but also the emergence of eukaryotes. So there is a lot of stuff going on in that period of time, and understanding the phosphorus cycling in that period is important. So let's. Oh no, it isn't. Okay. So let's start with this this equation for respiration or photosynthesis. The idea here is to show that when we stimulate organic matter production, primary production, then we may end up with a high flux of organic carbon to the sediment, and this is considered as a source for atmospheric oxygen. So this works well for the Phanerozoic, and I will have two main questions today that I don't really have the answer, but I have ideas of maybe how to look at this. The first question is, how long did this occur? This relationship between organic carbon, oxygen, and phosphorus, because of course you need phosphorus nutrients to stimulate this primary production here. So how far back do we have this close relationship between these three systems? If you look at today's biomass, there is this stoichiometry, so-called the Redfield ratio, of approximately 106 atoms of carbon for 1 atom of phosphorus. So this is what you would have today. And a second question that I'm asking myself is, or some question that I find very intriguing is, how old is the Redfield ratio? For how long has it been the case that we have this stoichiometry? Why is this the case? And obviously, when I started this this research, I had some hypotheses about these two questions, but they have evolved, they have changed through time, when I was investigating these systems. So let's start to try to look at these questions more in detail. So if we look at, so this is obviously the atmospheric oxygen curve here from 3.5 billion years ago till the modern. We are focusing on the Proterozoic today with a great oxygenation event here. And what we can see from the dual ion formation records and the phosphorus records is that there seems to be an anti-correlation between the two depositions. So it seems that when we deposit iron-rich sediments, we don't have iron-rich phosphorite-rich sediments, and vice versa. So there seems to be a link between the evolution of the redox chemistry of the oceans and the deposition of phosphorites. So how have people tried to reconstruct this Proterozoic cycling of phosphorus? Well, looking first at ion formations, because there are iron-rich chemical deposits which have the propensity, the iron oxyhydroxides that form in the water column have the propensity to scavenge phosphorus out of the water column. Meaning that if we measure the iron to phosphorus ratio in these formations, the idea is that it scales linearly with phosphorus in the seawater. So to support this, if you look at hydrothermal vents and sediments today, you would see that you have this very strong correlation here between phosphorus and iron. Why is this? Well, when you have iron coming out of hydrothermal vents, it quickly oxidizes with the oxygen present, and this strips out phosphorus out. So hydrothermal vents are not really considered a source, but rather a sink of phosphorus today. And if you look at ancient sediments, in that case, Neo-Archean sediments, in the red or orangey colors here, you can also see a strong correlation suggesting that indeed you have this scavenging of phosphorus occurring, and that we may use this iron to phosphorus ratio in these ancient chemical sediments to reconstruct phosphorus levels. However, there are things to be aware of here, is that first of all, if today's hydrothermal vents precipitate iron oxides and oxyhydroxides straight away, scavenging phosphorus, it may not have been the case in the Proterozoic, because the oceans were anoxic. The deep ocean was anoxic and ferruginous in these times. So we may not have this quantitative removal of phosphorus back then, but I will go back to that a bit later, because that we can look at experiments to look at this. So looking at this ratio, nevertheless, 14 years ago now, Noah Planowski and his co-workers and other groups have suggested that, or have reconstructed quite high level of phosphorus, or at least suggested that phosphorus was not a limiting nutrient in most of the Precambrian. Looking at this iron over phosphorus ratio in the past. Other suggestions of high phosphorus levels have come more recently, looking at carbonate-associated phosphorus, so in ancient carbonates this time, as a proxy for phosphorus levels in seawater, but also with the fact that in iron formations you have disseminated apatite, sometimes nanocrystalline, and that this suggests that this would suggest that you have higher levels of phosphorus than previously thought. Now, another thing regarding iron formations is that bear in mind that they are not continuous throughout history. I showed you earlier they stopped depositing in the Proterozoic. So and their formation is still highly controversial, at least very debated. The precursoring minerals for their formation are not certain, so let alone the behavior of phosphorus during diagenesis and or metamorphism. However, there was this nice study by Shad in 21. Oh, there is no label here, sorry. So I will explain that to you. Basically, they started with phosphorus-doped ferrihydrite, which is this reddish color here, and they stimulated diagenesis with and without organic carbon. And looking at the end product, so I think, if I remember well, magnetite is that black color here. Vivianite is in blue. And basically the essential results from this study was that less than 10% phosphorus is recycled back to the solution when you stimulate the diagenesis of these iron-rich sediments. And this is presumably because these sediments are full of iron oxides and oxyhydroxides, which are very good at stripping down and keeping phosphorus in the sediment. So to summarize this little introduction about what evidence would we have for higher levels of phosphorus in the Proterozoic? Well, the fact that hydrothermal vents could be considered sources of phosphorus, an additional source. The fact that you have disseminated apatite, also this new proxy, the carbonate-associated phosphorus, all seem to tend to suggest that higher levels of phosphorus were available in Proterozoic oceans. Let's have a look now at the record that you get from black shales and, well, from shales in general, because they constitute a much more continuous record throughout Earth history. They are everywhere. Their diagenesis is much more understood, simply because they are in brackets, normal sediments. And looking at, so here you see examples of phosphorus deposits in shales. When you look at the bulk content of shales, of phosphorus in shales through time, well, this is a huge compilation initiated by Christopher Reinhardt and then Noah Planavsky, who suggests that you have low phosphorus content throughout most of the Earth history. And then you have a change point around 750 million years ago or so with higher phosphorus contents in the sediments. And then they suggested that this is good evidence that phosphorus levels were low in the Proterozoic. Now, although we have a continuous record for these shales, measuring total phosphorus, bulk contents, doesn't tell us neither much about the fluxes of phosphorus into the sediment and the flux out of the sediment via recycling to the water column during diagenesis, for instance. So this is where we can combine that with experiments, etc., to address this. Something that is interesting is that, so why do we have these low phosphorus contents through the Proterozoic in these anoxic and iron-rich sediments? Luderi proposed a very nice scenario whereby you have vivianite such formation through a range of iron concentrations. And vivianite is an iron 2 phosphate that would titrate phosphorus out of the water column continuously, because you have this endless source of iron that is available to strip out phosphate. So in that scenario, you don't need to have massive phosphorite deposits, because then you would have this continuous and discrete precipitation of vivianite. And another interesting thing is that vivianite can transform into apatite through time. So I make the link here with the discovery of disseminated apatite in ancient iron formations. However, this transition from vivianite to apatite is still to be investigated. It's not well known yet. So there is still more work to do. Something that was interesting from the previous slide is that there seemed to be no real effect, although, well, this is a logarithmic scale, obviously, but seems to be no real effect of changes in phosphorus contents through the Proterozoic, although there seem to be changes in the redox chemistry of these oceans. And one could assume that if you have changes in the redox chemistry of the oceans, then you would have changes also in the cycling of phosphorus either into or out of the sediment. And this was exemplified by a study on the Cretaceous by Christian Mertz, who showed these variations between euxinic conditions, so anoxic and sulfide rich in white here. And in gray, you have this ferruginous condition, so anoxic and iron rich. And you see different behavior for phosphorus. Indeed, under euxinic conditions, you have sulfide that will dissolve the oxyhydroxides, releasing back phosphorus to the water column. So it's not stored in the sediment. And you can see that the phosphorus over aluminum ratio here is very low in this euxinic environments. Whereas under ferruginous conditions, you don't have this sulfide to dissolve iron oxyhydroxides, and phosphorus is indeed trapped into the sediment, making these peaks in the phosphorus over aluminum ratio. If you now look back at the organic carbon or the total phosphorus, here in the dashed line, you have the red-free ratio, and you see that these euxinic conditions are very good at releasing phosphorus back to the water column, but also of this propension to preferentially release phosphorus over carbon. So you will have more, well, higher ratios of carbon over phosphorus in these euxinic environments, whereas under ferruginous conditions, you will have lower ratios because you keep the phosphorus down. So, to summarize, we have, for this little part, we have elements that suggest lower phosphorus levels as opposed to what other evidence that I showed you earlier. The fact that phosphorus contents are below the average shale values throughout the Proterozoic, that you have rare phosphorite deposits during that time. Vivianite saturation may explain why you have a continuous removal of phosphorus from the seawater. And what I didn't really speak about was the more limited organic carbon degradation under oxic conditions. Today, most of organic matter is degraded within the water column of oxic waters, releasing back the phosphorus to the water column. Obviously, in the Proterozoic, this was not the case. So this limited organic carbon degradation may have also stripped down phosphorus down to the sediment. So how do we further look into this to try to answer to these two questions? How long has the relationship between phosphorus, carbon, and oxygen operated? And how old is the red-fill ratio? So my approach as an experimentalist is usually to make experiments, of course, and bring that into the observations in the modern environment. So in particular, looking at euxinic stratified lakes, but so this is Lake Cardaño here. But we tend to look at sulfidic lakes that will be low in sulfate, and this is important because this is probably what we would have had in the Proterozoic. So within these anoxic waters, you have different forms of iron minerals, mainly maquinawite, which is the first precipitate. The first iron sulfide precipitate to occur. You also have greigite, of course, pyrite, elemental sulfur. Then you have other lakes that are ferruginous, in which you can form green rust particles, which are mixed iron 2 and iron 3 hydroxides. You also can have ferrihydrite and other oxides, including magnetite. You can have siderite and vivianite forming in these ferruginous lakes. And I put here greenolite in brackets because you really need dissolved silica to form these minerals. And this may have occurred, of course, in the Proterozoic, but in modern lakes, it's a little bit more tricky to find. So, and we try to combine all these things. So we'll talk first about ferruginous conditions and then euxinic conditions in a bit. So how did we, with my colleagues, investigate ferruginous anoxia? Well, this is the work by my previous PhD student, Jenny Thompson, who studied Lake La Cruz in Spain, which is anoxic and containing dissolved iron. I put iron rich here it's a little bit tricky to say it's iron rich because it's not really iron rich. There is dissolved iron, but it's not comparable to other systems. So if you go down, you have oxic conditions up to about 10 to 12 meters, and then you have iron, which will start to iron 2, which will start to be measurable in the water column. So this is what we see here. Here, this is the water depth in meters. Here, this is the redox line in red, and you can see that below the redox line, you start to have this accumulation of iron 2, dissolved iron 2. And you also have a flux of phosphorus coming from the sediment here, because you see that is coming there, and this is presumably because of this sulfide recycling in the sediment. What's happening with the particulate flux? Iron 2 particles form below the redox line, and this is because of the reaction with the upwelling iron 2 here. So as you see, it's not because you have iron 2 in the system, like dissolved iron 2, that you cannot have iron 2 particles. And these are very good at stripping down phosphorus. So the idea that because you don't have oxic conditions in the full ocean, in the Proterozoic, doesn't mean that you don't have these particles, these oxyhydroxides forming, raining down the water column and stripping down the phosphorus on its way down. You can see that particulate phosphorus in these water columns also forms, and this is obviously because you have this particulate iron 2 flux here forming. So now what's happening in the sediment? Jenny performed both iron and phosphorus speciation on these sediments, and most of the iron speciation is dominated by these iron 2 phases, which are not sulfidized. So sulfur are in gray here, so pyrite and ABS. So this is enough to recycle the phosphorus back to the water column. Now we saw this little flux of phosphorus, but most iron is made of iron 2 phases here. And we want to understand which phases, of course. Phosphorus is mainly bound with iron species, which you can see in red, and which you can compare with this iron 2 species just next to it, and also organic phosphorus. So when we want to understand the diagenesis of these iron-rich phases and iron phosphates, this is where experiments come along. In particular, I'm interested in the formation of vivianite, of iron 2 silicates. So we speak a lot about granulite, but really what happens when you mix iron 2 and silica is you form this amorphous iron 2 silicates with some starts of some crystallites here, but you really have a large amorphous mass here. And you also can form green rust. So this is the other mineral that I'm interested in. And here, this is some work by Jeffrey Paris, who came from GFZ to do some experiments in my lab, where he looked at the competition between green rust and granulite at 25 and 90 degrees, reproducing seawater, Proterozoic seawater, and seeing what happens with phosphorus here. So basically, we have both minerals forming at lower and higher silica contents, but the mineralogy indicates clearly that you have you can have both granulite and green rust in these experiments. And the interesting bit here is that you have high levels of phosphorus removal. This was done over a month. And you see that you have between 60 and 80% of phosphorus that is removed from the experimental seawater. You also see in the darker blue color there that when you have more granulite, you have less removal. So it seems that green rust is very powerful at stripping down phosphorus from these water columns. So kind of a conclusion that we can have here is that indeed the formation of these iron-rich minerals are very good at removing phosphorus, and that it seems that over 30 days at 90 degrees, phosphorus is majoritarily in the sediment phase. Another PhD student that I had in Leeds, Yijun Zhong, investigated the competition, this time between green rust and vivianite. So we saw that green rust can take a lot of phosphorus, can remove a lot of phosphorus, but so what's the competition with vivianite here? When does vivianite form? So he looked at two stoichiometries of green rust only and vivianite only, and then varied the stoichiometry of the phosphorus to iron ratio to represent some kind of natural environments, Lake La Cruz, Lake Mazzano, Lake Pavin, and Lake Cadagno, and observed that you have green rust forming, only green rusts, when the ratio of iron to phosphorus is less than 1:30. When you have stuff that is starting to be higher than that, you have a mixture of both. And in fact, it suggested that, so this is the mechanism to form green rust, whereby you have this brucite-like sheet structure with iron 3 hydroxide and iron 2 hydroxide layers. In between, you can fit some anions, in that case sulfates, but you can also accommodate phosphate within this as interlayer anions. And when you have an excess of phosphorus, then you start to dissolve the green rust, and the iron 2 hydroxide that accommodated the phosphate recrystallizes into vivianite, and this is when you start to have this vivianite forming with iron 3 hydroxides. So the combination of these observations and these experimental data tend to strengthen the point that ferruginous conditions are good to fix phosphorus in the sediment phase because of the reactivity of these iron 2 particles. Then they can be in competition with each other, but the result is similar in all ways. That is, you have a fixation of phosphorus in the sediment. So now let's have a look at what we have during euxenia, during anoxic and sulfide-rich conditions. So we move to Lake Cadagno in Switzerland, and here it's relatively similar to what happens in Lake La Cruz. After 10 meters, you don't have oxygen anymore in the water column, and you start to build sulfide this time in the water column. You have a lot of purple bacteria, purple bacteria, the redox cline. So the color, I don't know, well, you probably don't see that well here, but the color of the water when pump it out is totally pink. It's very nice. And then we observe the building up of dissolved sulfide below 12 meters or so, or 10 meters. Just before this, we observe a peak in particulate iron 2. And this is because the iron 2 that is released from dissolution of iron oxyhydroxides here quickly reacts to form some kind of minerals at the redox cline. And this is accompanied by another peak in particulate phosphorus. Again, this makes sense because we just saw that these iron 2 minerals are extremely good at stripping down phosphorus. Below the when you start to have sulfide forming, then you dissolve these phases and you start to have a buildup of phosphorus in the water column this time. So experimentally, when we look at this area here, that this redox cline here, where you have the partial oxidation of sulfides, we can look at what happens experimentally. So here, this is an experiment starting with Makina white only, and here you can see these beautiful platelets of Makina white. So this is typical from when you precipitate Makina white in an experiment. This is how it looks, and when you partly oxidize it, you will start to form other minerals, such as these hexagons, which are green rust particles. So we see that at this redox line between euxinic conditions, then you start to partly oxidize makinawite into green rusts, which are these mixed iron 2, iron 3 oxyhydroxides, which explains why you have this peak of particulate iron 2 combined with the phosphorus here, because as I just showed you, green rust is capable to accommodate quite a lot of phosphorus. So Eugene also looked at the speciation of phosphorus in the sediments. First of all, looking at, so now we are in the sediment pile, not in the water column, looking at the organic over organic carbon over organic phosphorus ratio. And as I suggested earlier, because of sulfide, you have this recycling of phosphorus back to the water column. So the ratio is very high. Here you have the Redfield ratio. But it goes down, and it goes down here because you have, in depth, you have the consumption of sulfide. You have an excess of iron, which and this lake is quite low in sulfate in the first place. You don't have too much to fuel sulfate reduction. So you have a consumption of sulfide, which is reflected here in the speciation of phosphorus, because here you transition from a part of in the first sediments of the first centimeters of the sediment, where you have a speciation dominated by organic phosphorus, whereas if you go deeper, you have a speciation dominated by iron phases. And Mark modeled this. This could correspond to a mixture of things, including vivianite, magnetite, siderite, and green rust. So, unlike for what we had for ferruginous conditions, what we seem to see here is that as assumed, I would say, euxinic conditions are very good at recycling phosphorus back to the water column, but this is nuanced with vivianite formation or other phase formations when sulfide is limited in the sediments. So this is to be taken into consideration as well, that in the Proterozoic, if you don't have too much sulfate available for sulfide production, then maybe this recycling of phosphorus can be diminished, and we will see if we see that in the rock record. So this brings me to the third part of my talk, which is how can we reconstruct the redox cycling of phosphorus in the Proterozoic, and how do we make sense of all of this data that we have from the iron formation record, but also from the black shales. So you remember you have this oxygenation history, and the oceans remained anoxic for most of the Proterozoic, with oxic waters restricted to the shallow waters. And here, what is assumed is that you have this the formation of sulfide in the water column is quite restricted. It's restricted to continental margins, where you have this flux of organic carbon, so arriving to the continental margins and stimulating a sulfate reduction. The amplitude, the widespreadness of these euxinic conditions is, I would argue, probably overestimated. We don't really know that yet, but this is probably very limited anyway to the continental margins. And yet it has evolved through time. We have plenty of data suggesting that you have more or less of these euxinic conditions through time. But as I said earlier, in the black shale content, bulk content, you don't seem to see a big change here in this when the redox chemistry changes. Although you would expect that if you have these euxinic settings, you would have more recycling of phosphorus to the water column, more primary production, a higher flux of organic carbon, diminished carbon over phosphorus ratios, and a potential source of oxygen to the atmosphere. Whereas under ferruginous dominated conditions, you have a stripping down of phosphorus to the sediments, diminished carbon over phosphorus ratios, and a lesser flux of organic carbon burial under this ferruginous condition. So can we see something like this in the deep past? And in order to do that, and to not look at bulk sediments, but trying to differentiate between detrital and potentially bioavailable phosphorus, Jenny Thompson developed these protocols for phosphorus and iron speciation applied to ancient sedimentary rocks. Obviously, the point here is to be able to distinguish between available or phosphorus that is potentially available and phosphorus that never was. So that I call here detrital phosphorus. And to be able to say that, you need to have checks, and in particular, the detrital phosphorus needs to be correlated with other detritic elements such as aluminium or titanium, because this guarantees you that the detrital, the phase that you extract as detrital phosphorus is indeed detrital, and it's not recrystallization of other phosphorus phases such as orthogenic apatite or iron oxides, etc. So this means that this doesn't work all the time, and you need to interpret these results only when you have done these checks. Or otherwise, I think that this is pointless to do really. So this method, it can be very powerful. But sometimes you just end up with stuff that you cannot interpret because you have too much recrystallization. So you have to bear that in mind. So we looked at this for different basins. The Huainan Basin, which is 1 billion years old. The Taodeni Basin, which is, the Huainan Basin is 1 billion years old, and it's dominantly ferruginous. There is very low sulfide in these sediments. And two other basins, the Taodeni, which is 1.1, 1.2, and the Anamiki, which is 1.8 billion years old, and that are mainly sulfidic. So here, what I do is I look at the carbon over phosphorus ratio, but doing the iron speciation, the phosphorus speciation. So looking at organic phosphorus and reactive phosphorus. So this is not total phosphorus. This is total phosphorus minus the detrital phase that I just showed you before. What you see is that in these ferruginous conditions, if you look at organic phosphorus, it follows very well the rate-fair ratio. So that answers, and that's 1 billion years ago. So that answers partly the question, how old is this stoichiometry of the rate-fair ratio? Well, at least it seems that in this 1 billion years sediments, it was already there. Like otherwise, it would be extremely an extremely weird coincidence that we have everything plotting on the rate-fair ratio. Whereas if you look at these euxinic sediments, you see evidence for recycling of phosphorus back to the water column, as expressed here by this higher values, higher C over P values. So looking at this, I went back to the literature to look at what's happening earlier, and of course, you have to make sure that you can compare things. So you need to look for sediments where the phosphorus speciation has been done and where you have evidence that detrital phosphorus is not in fact recrystallized orthogenic phosphorus. And so of course that filters a lot of data. But I went back to the paper by Alcott in 2022, where they looked at approximately 2.5 billion years old shale and iron stone. So we are at the great oxygenation event. And what we see is that in the iron stones, we have something that is equal or below the rate-fair ratio. And in the shales, where there is sulfide, you have much higher ratios. You have something that is much higher than the rate-fair ratio. Here in blue, I also put the line which corresponds to a ratio of 300 to 1, which has been suggested to be the ratio for which organisms take phosphorus when they are phosphorus limited, when there is a stress. And I did that to compare what we have in the later Proterozoic. And what you can see here is that you seem to have more vigorous recycling of phosphorus at the time of the great oxygenation event, rather than later in the later mid-Proterozoic, where this recycling seems to be much less vigorous. So you can argue, well, this is, you know, we need more and more data, but it seems to me like wanting to answer to the first questions I was asking earlier. It seems to me that we tend to see this relationship between phosphorus, carbon, and oxygen way back to that time. Now, about the antiquity of the Redfield ratio, I'm still amazed and I'm still surprised that we see things that this stoichiometry hasn't changed for that long. And of course, if you look at bulk, so the bulk contents again. So this, I just took the compilation by Reinhardt, and I looked at the TOC of a P ratio through time. Here's the Redfield ratio. You don't really see much here, but if you look at the Phanerozoic median value, you have something that is spot on the Redfield ratio, and that it diverges afterwards. And you may ask, okay, it diverges because we have more degradation of organic matter in these old sediments, or it diverges because you have less or more or less phosphorus available. So what I suggest to try to answer that is to try to consider data that were of shales that were deposited under ferruginous conditions. So this is in order to avoid massive organic carbon degradation, and for sediments in which you have very, very little sulfide. So of course, there is not a lot of data, but this is a suggestion of what we could look at. And here I filtered all the shales for having less than 0.05 weight percent sulfide. So there is nothing. And they are all deposited under ferruginous conditions. And what we see for these shales is that they are either on this Redfield stoichiometry or for the Doyle data. So this is 1.4 billion years old. So yes, I think so. Now I'm not sure if it's 1.4, 1.6, but you can see anyway that the data are plotting between this phosphorus starving ratio and the usual Redfield ratio. So to answer to the question I was asking earlier about the antiquity of the Redfield ratio, I think, well, we could look into that, but I have the impression so far that we have this kind of stoichiometry already operating in the Mesoproterozoic. And who knows if it was there earlier. So for me, that's a big surprise, because when I started to think of these questions, I was thinking, well, the Redfield ratio must have changed all the time. Like organisms must have taken whatever there was. And it seems that at least until the Mesoproterozoic, it seems to be a robust number. Right, so I think I'm arriving at the end. To summarize very quickly, if we look at all the data all in all so far, I still think that imagining low levels of phosphorus in the Precambrian, at least until approximately 800 million years ago, are the most plausible situation. The relationship between phosphorus, organic carbon, and oxygen seems to have operated back in the Precambrian. And we can see that even back to the great oxygenation event. And we have limited but growing evidence from organic carbon indicated that the Redfield ratio wasn't changed for at least 1.8 billion years. Thank you very much.
Okay, thanks, Roman. We'll go ahead and switch to asking questions, the question session. You can ask either by typing it into the chat and I can read it for you, or you can find the reactions button at the bottom of your screen and click raise hand. Let's see.
I forgot to thank all the people I work with, by the way.
Oh, you can do that now if you want.
Sorry, guys.
Okay, so the thanks slide is up. So recognize people. All right. Ava, please, Ava Stoican has a question. Please go ahead.
Yeah, thanks for your talk. It's interesting. So I was wondering what your take is on the carbonate data that suggests higher phosphate levels in the Proterozoic. What do you think is going on there?
Well, well, it's a new proxy, so we'll see. We'll see how. I'm very interested to see what the later results will be. I'm also thinking that what carbonates are we looking at? Are we looking at micrites that are diagenetic and that there have been, the oxidation of organic matter, because then we would have a release of phosphorus in that aspect also. And so I'd be just careful in what exactly we are analyzing when we say carbonate associated phosphate, basically.
Could it also be, sorry.
Sorry?
There's one could it also be some of this iron chemistry happens within carbonate as well? So if there's any iron in the carbonate, could that also play a role, I guess?
Yeah, sure.
Yeah.
No, totally.
Yeah, thanks.
All right. Thanks. Adriana Rizzo has a question in the chat. Says, great talk. Could you say more about what your interpretation of carbon phosphorus ratios below the Redfield ratio would be?
Oh, well, obviously, diagenesis occurs. When you imagine organisms falling down the water column with let's say that they have the exact Redfield ratio, then of course you have other sources of phosphorus to the sediment, and that's with pool changing during diagenesis, then you can increase this, you can decrease, sorry, the carbon over reactive phosphorus ratio. So this is the way I interpret that. What the phosphorus speciation allows us to do is to differentiate between what is total, what is reactive, and what is organic. And of course, the other possibility is that you have more organic degradation during diagenesis, and that the phosphorus that is liberated during that process is then straight away fixed by other iron oxides, etc. So if you have organic carbon degradation within these ferruginous or iron-rich sediments, it may be that the phosphorus cannot go back to the water column, but instead is fixed in the sediment, and that would decrease the organic over reactive phosphorus ratio. I hope I was clear. I know.
Yeah. All right. Thanks, Roman. All right, dude, let's see. We don't have hands up right now. We'll give a few moments to see if anybody would like to speak up.
Yeah, sure.
Paul would like to speak. Go ahead, Paul. Yeah, you're still muted. Okay, there you go.
The first biomineralizing organisms occurred around 810 million years ago, and they formed, these are the scale microfossils, and they formed hydroxyapatite. So what is the significance of that?
So do they form, are you implying that this is the primary mineral they form, or this is?
Yes.
Yeah,
yes.
Okay. Well, that's so, and you say 800, yeah?
Well, the first biomineralization utilized hydroxyapatite to form skeletal components.
Yeah,
So
yeah,
this
yeah.
is a rather extravagant use of a limiting nutrient.
Yeah, yeah,
What
yeah.
does that tell you about phosphorus content of seawater at that time?
Yeah, well, and that's also exactly, well, when, or more or less, when you start to have this higher phosphorus contents in as a bulk. So this is where people seem to suggest that you have higher contents of phosphorus of phosphate in seawater at that time. This is where you have this change point, and which is also coincident with biomass that starts to be more eukaryotic dominated. So yeah. So yeah,
Part of that was
sorry.
part of that was because of the high phosphorus content of cryogenian iron formation.
Yeah, yeah.
And of course, I think that the reason there was a high phosphorus content of seawater during snowball Earth was because of light limitation. So there was very little uptake of phosphorus because the oceans were dark. So I have a completely different interpretation of that than Noah Planavsky.
Yeah. Okay. Nice.
So I don't think that the cryogenian iron formations tell you very much about the ambient phosphorus content of seawater under non-snowball conditions.
Yeah, that's a good point.
I mean, the polar oceans today have high nutrient content because of sea ice.
Because sorry,
Well, the polar oceans have limited uptake of phosphorus because of sea ice during much of the
yes. Sorry, I couldn't hear. Yeah,
naturally, the nutrient content in the polar oceans is quite high because of but it's because of lack of uptake. Because the limiting nutrient isn't phosphorus, it's sunlight.
What you seem to see also in the Jacaran is that you seem to have much more sulfide in sediments, and then if you have that, of course, you have more recycling than what you had in the previous time period. So
I find the biological, the evidence for hydroxyapatite biomineralization more compelling as an argument for high phosphorus in seawater in the Latonian than other evidence.
Yeah, I think that's a good point.
Cool. Thanks, Paul. Greg, you have your hand up. Would you like to go ahead?
That was fascinating. I'm still trying to come to grips with it because I don't know if you know, but I did a compilation published a couple of years ago of phosphorus depletion from paleosols back 4 billion years. And yes, there's two big steps in phosphorus depletion in paleosols, one at the Great Oxidation Event and one in the Neo-Proterozoic, almost an order of magnitude, both of them. But what you seem to be telling me is that's not seen in the ocean because of this, I guess, seesaw between euxinic and what you call ferruginous and I would call ferrous. It's kind of evening itself out and getting back to red-fill ratio. What are your thoughts about that? I mean, if the delivery differs so much, why is the outcome so constant?
Well, I didn't say that. I think that what I said is that it seems to be muted if we look at the bulk phosphorus content in shale. What I think is that when you look at a speciation of phosphorus in the shales, you do see a difference between what is potentially bioavailable and what is only detrital. And that you see an evolution of that. This was a bit my way to say, well, the bulk shale content don't really reflect the whole complexity of phosphorus availability at all. It is true that the older, because of this recrystallization of orthogenic phosphorus, you're always in most sediments. Basically, the speciation will not be possible in most sediments, I think, because you have high amounts of recrystallization, and then you can't really interpret that. But when you can, that is when you can really see detrital, differentiate between detrital phosphorus and the other phases, then you do observe a difference, at least in bioavailability. So.
Yeah, I'm still trying to wrap my head around it. There's this big signal coming from the land, and yet it's flattened out in the ocean. That is intriguing. Anyway,
right, thanks.
thanks a lot. I'll keep pondering.
Yeah. I mean, I don't have an answer either. It's
Fascinating. Thank you so much.
Yeah, Roman, I guess I've been, that's what you presented is a lot to think about, and I've sort of been trying to chew on it. I'm thinking about the interaction between minerals in your modern euxinic and ferruginous basins with scrubbing phosphorus from the water. And it seems to me like everything kind of does to some degree, these iron minerals, everything kind of does to some degree scrub some phosphorus. You showed specifically the difference between the greenolite and green rust. But it seems like there's also a scrubbing of sulfides as well. Sulfide formation, would that be correct? Does sulfide pull down phosphorus?
No, well, sulfide would dissolve most of these phases, and including vivianite, and then you create a flux of phosphorus back to the water column. So
right, okay,
eventually,
so
if that reaches the photic zone, then you can stimulate primary production. The sulfidation of these phases, well, in particular, so vivianite now is well understood. Greenolite is much less so. So there are still a lot of work to understand phosphorus dynamics when you have greenolite entering sulfidic sediments, I think. This is not well understood at all. Of course, none of these phases can be considered primary. So you have to bear in mind that so what we do when we do experiments is we see what is happening at time t, so in situ, but then all of this will be potentially reworked afterwards. This is why these diagenetic experiments are so important. Nevertheless, yes, the non-sulfidic phases seem to be quite good at removing phosphorus in different amounts. And more importantly, when they transform during diagenesis, for example, vivianite, the Lou Derry's hypothesis about vivianite, that was very stimulating because, I'm making that in the lab. I want to see what happens. But then we don't see vivianite in most BIFs. There is usually no vivianite. Vivianite is very difficult to say in ancient shales. However, vivianite transforms into apatite, and that keeps the phosphorus. So yeah, these things combined, they will start to make sense,
So the other thing then, looking at that difference in the percent of phosphorus associated with the experiment with higher dissolved silica, and how that was that had lower percent phosphorus removed. The other thing that we kind of see changing, around these Neoproterozoic times and 800, we sort of see we don't know exactly when, massive, a lot of silica biomineralization was starting, but, early sponges could have, silica sponges could have started around then. I mean, do you think that this drawing down of silica from the oceans could have been impacting an important factor in phosphorus?
Yeah, so interesting. I actually, I'm interested in high silica contents to reproduce that time before the silica removal by these things. But it's a good question. I didn't think of that. Here, the only thing that we focus on is silicates that are rich in iron 2. And iron 2 decreases when you go towards the terminal adjacent. So potentially it has an impulse. Yeah, potentially yes. Then also what we see is that it's not the best either at removing phosphorus. It's quite good, but I think the fact that you can incorporate phosphorus within the interlayers of green rust, that makes a difference. And this is why you have more removal of phosphorus than with greenolites. But as I said, both can occur. There is no reason why we should think that a Proterozoic ocean could not have had both phases. And they co-occur at least, and they are both quite good at removing phosphorus. So what happens when you have these biomineralizations? Well, it's a good question.
All right, thanks. While I still have you, I'll ask you one more. So, and this is more just about your experiments when you're getting iron silicate minerals. Do you really only usually get these, these silicate minerals that don't have aluminum, or do you occasionally get aluminous iron silicates, or does that have to do with maybe the solution you're using? Can you
well,
maybe comment on aluminum?
I decide what I want to precipitate.
Oh, okay. I see.
It
All right.
entirely depends on what I put in the system.
Got it.
Here, what
Okay.
I showed you is there is no aluminum.
Okay, got it. Thanks.
Of course, we can evolve towards more and more complex mineralogies and stuff like that, but then it becomes increasingly more difficult to understand what does what. So yeah, first we start with that, and then we'll see.
Yeah. Okay, cool. Thanks. I'll leave a few seconds in case anybody wants to speak up. Go ahead, Paul.
Yeah, it looks like there isn't silica biomineralization until the base of the Cambrian. It looks like the ediacaran sponges seem to make spicules out of organic matter mainly, and they're very lightly mineralized. And there's also genetic arguments for the early demo sponges being not biomineralized.
Got it.
Okay.
I'll have to read up more on that. All right, Ava, you wanted to speak up again? Go ahead.
Yeah, so I have a biology question. This might be a bit off topic, but so I wondered, so in the Proterozoic, of course, we had mostly prokaryotes dominating productivity, and today we have eukaryotic algae that kind of define the red field ratio. And so I wondered if you have any knowledge of, I guess, the enzymes that regulate the red field ratio, or if it actually if it was conserved over billions of years, if that tells us anything about the biology.
No, not really. The thing is that I would have my presupposition was that it was not it couldn't. I thought it could not be constant because we come from a world that is prokaryote dominated to a eukaryotic one, and I thought, well, there are high chances that early organisms were just taken whatever there was, and that it evolved with biology simply. So I'm the first surprised to see that in these sediments where the deposit in ferruginous conditions and where you have virtually no sulfides, you can preserve it when you analyze the organic matter, and so I'm the first surprised to that. Again, it's questions that are raised now, but I don't have answers to this. I'm happy to
Yeah, thanks.
to hear what you think. Maybe like.
I don't know. I'm not a biologist, and I'm not aware of any particular enzymes that control this ratio. There are probably lots of them. So yeah, I guess I agree that it would be very surprising.
Okay. All right. Well, we're just past the hour. So, Roman, thanks so much. That was really interesting talk. Yeah,
thank you very
and
much
thanks.
for the invitation.
Yeah, and thanks everybody for participating. We'll see you guys around. Take care.
Bye-bye.
Thanks, Romain.
Yeah. Oh, thanks. Bye-bye.